process of DNA replication involves 6 enzymes consisting of helicase, topoisomerase, primase, DNA polymerase III, ligase, and DNA polymerase I.
DNA stands for deoxyribonucleic acid. The structure of DNA is represented by a double helix structure. DNA is made of nucleotides, which consist of deoxyribose sugar, phosphate groups, and nitrogenous bases. There are 4 different types of nitrogenous bases present in the DNA, including adenine, thymine, cytosine, and guanine. In accordance with Chargaff's rule, adenine always pairs with thymine, and cytosine always pairs with guanine. Therefore, the amounts of adenine are always equivalent to the amounts of thymine, and the amounts of cytosine are always equivalent to the amounts of guanine.
DNA replication occurs at origins of replication, where proteins recognize a sequence of nucleotides. While bacteria only have one origin, eukaryotes have up to thousands of origins. The separation of the 2 DNA strands occurs when the enzyme called helicase opens up the replication bubble. Replication goes in both directions of the strand until the whole DNA molecule is copied. Single-strand binding proteins coat the DNA around the replication fork to prevent the rewinding of DNA.
Topoisomerase is an enzyme that works ahead of the replication fork in order to prevent supercoiling or rewinding of the DNA strands.
Uncoiled DNA is anti-parallel. DNA polymerases can only add nucleotides to the 3' end and therefore only builds in the 5' to 3' direction. The leading strand runs in the 5' to 3' direction, whereas the complementary strand runs in the 3' to 5' direction since they are anti-parallel. Since nucleotides can only build in the 5' to 3' direction, the leading strand can be synthesized continuously away from the replication fork. The lagging strand, however, is synthesized in fragments towards the replication fork, but still builds in the 5' to 3' direction.
DNA polymerase cannot begin to attach complementary nucleotides to the template strands until a primer is made. An enzyme called primase creates an RNA primer complementary to the old strand. From there, DNA polymerase III can begin to synthesize DNA in the 5' to 3' direction. DNA polymerase is able to check its work after it's done and fix any mistakes.
In the end, RNA primers are replaced by DNA nucleotides by DNA polymerase I. Circling back to the ligase, gaps produced after the activity of DNA polymerase I are sealed by DNA ligase.